I am Fan Chung. Today I'd like to present a synopsis of this study that has just been published in Allergy. I'm presenting this on behalf of the UBioPred study group and on behalf of our collaborators in Australia. The background to this study is that we know that late onset asthma is associated with more severe asthma and also with a number of comorbidities. And recently there has been some evidence that some cellular pathways, senescence pathways, is involved in severe asthma. Therefore, in this study, which is using the rich data of UBioPred, we applied two gene signatures that represent cellular senescence pathways. One is a SASP gene signature, which is the senescence-associated secretory phenotype, which represent the proteins that are released from cellular senescent cells, and some of the genes that are associated with the mechanisms of the release of those proteins and lipids. And the second signature relates to the p53 pathway, which is associated with cellular senescence. So we use those two signatures that were publicly available and applied them to the transcriptome of biopsies, bronchial biopsies obtained from patients with severe asthma, mild-moderate asthma, and healthy volunteers. And as you can see here, the enrichment scores for both gene signatures were increased in severe asthma compared to the other two groups. And within the severe asthma group, those that had nasal polyps had the significantly higher expression of those two signatures compared to those with no nasal polyps. And using multivariate linear regression, we also showed that the age of onset of asthma is significantly associated with enrichment scores of those two gene signatures, and also features of severity of the severe asthma, such as exacerbations, dose of maintenance oral corticosteroids, the presence of nasal polyps, and post-bronchodilator FEV1 were also significantly associated with both of those signatures. Interestingly, we found that serum pyroxine was also associated with those two signatures, particularly the p53 gene signature. Here is the spread of the concomitant expression of those two signatures in the cohort. And the one that is of particular interest is the group that had increased enrichment for both signatures, which is in that category that represent 46% of the severe asthmatics. And these asthmatics did show the highest exacerbation rate, lowest FEV1, highest daily OCS, and highest prevalence of nasal polyps compared to the other three groups. Finally, we determined which of those pathways that were important. And for this, we use the SASP gene signature, which is a large group of genes, as you've seen, and we clustered those genes according to protein-protein interaction networks. And we found 3 gene clusters. And of those 3 gene clusters, gene cluster 2 and cluster 3 were the most significantly increased compared to the mild-moderate asthma and healthy volunteers. And what do these 2 gene clusters represent? Well, gene cluster 2 represents genes associated with T cell regulation, with lung injury and repair, and with lung cancer, while gene cluster 3 was associated with steroid resistance, with immune response to pollution and airway wall remodeling, which are processes that are very much associated with asthma. We therefore conclude that we've shown translational evidence for cellular senescent pathways underlying the pathogenesis and severity of severe asthma from the ubipred cohort, and this is backed by analysis also of the Australian NOVA cohort. So the accelerated airway aging in severe asthma would involve those predominantly non-T2 pathways of T-cell regulation, airway wall remodeling, steroid resistance, immune response to pollution, and lung injury and repair. And therefore, we believe that targeting senescent pathways may be a way forward for tackling severe asthma. Thank you.